Great. It's really my pleasure to introduce Melissa, who comes to Stanford from Colorado State, where she was already sort of an extremely accomplished undergraduate protein chemist, researcher. And she, in the Bertozzi lab, has just really sort of been responsible for launching kind of multiple directions in the lab related to our utilization of bioconjugation chemistry, but also sort of the downstream immunological consequences of these sort of highly produced conjugates. And so Melissa, I think in the lab is to be credited with kind of bringing in sort of in vivo and mouse work and taking real translational sort of focuses with the bioconjugation chemistry the Bertozzi lab has pioneered. And so Melissa has won a number of awards. She's an NSF fellow, also a ChemH fellow, and has just really stimulated, I would say, a whole side of the Bertozzi lab related to protein engineering and protein synthesis that wasn't really sort of there before. And so I'm really excited to kind of hear her talk today and to hear sort of this really foundational story for the Bertozzi lab. And I think what will launch a lot of future projects, both in the lab and hopefully with a number of translational startup companies. And so with that, I hand it over to Melissa, and I think we're all in for a really exciting talk.
All right. So again, thanks for that introduction, Stephen. So today I'd like to talk about engineering antibody enzyme conjugates for enzymatic removal of sugars from cancer cell surfaces. So first, normal cell surfaces contain vast amounts of glycans, which can be found in the form of glycoproteins or glycolipids on the cell surface, or linked to proteins in the extracellular matrix. And combined, all of this makes up what we call a glycocalyx, or a sugar coating around the cell. And in cancer cells, there are many changes to the glycocalyx. And one in particular that I'd like to talk about today is very consistent across all cancer cell types and was first noticed about a century ago by researchers who discovered that cancer cells were almost ubiquitously more negatively charged than a healthy cell, a corresponding healthy cell. And in the 60s or so, it was discovered that the reason for this negatively charged cancer cell surface is that the cancer cells are decorating themselves with this negatively charged sugar called sialic acid, which I'm representing in cartoon form here as a purple diamond, also in the chemical structure shown there. And the term sialic acid describes a family of around 50 or so 9-carbon sugars that all contain carboxylic acid group, which gives it its negative charge. And they're typically presented on the terminal ends of a glycan. And the most common form in human is the one I'm showing on the right here, called N-acetylneurominic acid. And because of their broad overrepresentation on the surface of so many different cancer cells, researchers for a long time have been speculating about how cell surface sialic acids can benefit cancer survival, when in a cell culture dish, they seem non-essential on the glycocalyx to cancer growth and survival. And one key mechanism that has been elucidated in the past 20 years is this role for sialic acid sugars as a checkpoint ligand to a family of receptors called SIGLEX, or sialic acid binding Ig-like lectins. This has been further elucidated in the past 20 years, but it seems like when sialoglycoproteins bind to their corresponding SIGLEC receptors, which are expressed on a variety of immune cells, it can inhibit that immune cell from releasing cytokines or directly killing the cancer cell. And what's really interesting about this is that if we look at the cytosolic intracellular signaling domain of these SIGLEC receptors, they're quite homologous, many of them are quite homologous to the PD-1 receptor with these inhibitory tyrosine-based motifs. And this is interesting because, as we know, that the therapeutic approach to targeting and blocking this PD-1, PD-L1 checkpoint interaction has been really successful in treating melanomas in particular and other various types of cancers. But essentially blocking this immune inhibitory interaction can be a successful therapy. And so we're interested in bringing this sort of ligand blocking approach to prevent immune cell inhibition to the sialoglycan space. And one challenge, well, there are a couple of challenges associated with this. So, for example, PD-L1 is just one protein, and we can develop antibodies that bind to it and block it specifically. For glycans, it's very challenging to get antibodies that are good binders. Additionally, we're not really sure at this point what the most important sialylated proteins or sialylated lipids are that are acting as immune cell checkpoints. And additionally, it might not just be one or two of these sialoglycoproteins. It might be these sialoglycans represent a group of molecules with vast chemical diversity. So, for example, these sialic acid sugars can be linked in multiple different ways to glycans. These sialic acid sugars can also themselves be post-translationally modified with methyl groups or acetyl groups or sulfated. And we happen to know that the protein or lipid that these sugars are presented on the cell surface can affect how well they bind the Siglec receptor. So there's a lot of diversity, so it's very hard to block one of these or block all of them specifically. So a technique developed in the Bertozzi lab by previous members Hans Schau and Elliott Woods is to, rather than try to bind and block a very chemically diverse group of ligands, they instead conjugated a sialidase enzyme to an antibody. And that antibody, in their case, the trastuzumab antibody that targets the HER2 antigen on cancer cells. So the antibody gives this molecule some cancer cell specificity in binding to HER2 positive cancer cells. And the sialidase enzyme can then cleave off all of the sialic acids, this diverse group of ligands, so that we can better understand what these sialoglycans are doing in terms of the immune synapse. So Hahn and Elliott made a version of this molecule called trastuzumab sialidase conjugate, T-Sia, and they showed in vitro that they could enhance natural killer cell killing of cancer cells over antibody alone by also removing sialic acids from the cell surface. And when I joined the lab, I was really interested in moving this to in vivo models and really understanding what the role for sialoglycans were in immune evasion in vivo. And so I had to then, in order to take this model in vivo, I had to make some adjustments. So, for example, the sialidase used in the original version was a Vibrio cholera sialidase, and it contained two lectin binding domains that I couldn't engineer out while still making a stable protein. And these lectin binding domains gave it off-target binding and off-target desylation on cells that just possessed glycans, when really we were interested in having the targeting only and binding to cells only be mediated by the antigen binding of the antibody. So I needed to screen a panel of other sialidases looking for low off-target specificity, but still high catalytic activity and cleavage. And additionally, the linker in the original model was not site-specific and not stable. So those were two things I wanted to work on when I joined the lab. And I ended up developing what I call T-Sia version 2. So again, it's just a trastuzumab antibody, this time linked to a different bacterial enzyme. It's very stable, and it only contains the catalytic portion, so thereby reducing off-target effects. And it's from Salmonella typhimurium. And I also engineered a site-specific and stable linker. So I'll talk a little bit about the linker first. Here is an overview of the bioconjugation strategy I use, which I'll go through step by step. And critical to this whole process was the need for a bioorthogonal handle so that I could site-specifically react to my antibody in a way that wouldn't interfere with the antigen binding site or FC binding or immune cell binding site of the antibody. So in this case, I generated an aldehyde group using the aldehyde tag or smart tag technology. So this originated in the Bertozzi group and has been improved greatly by David Rabuka at Catalent. But essentially how this enzymatic bioconjugation works is all you need to include is a 5 amino acid consensus sequence of cysteine, any amino acid proline, any amino acid, and R-arginine. And if you can express this inside the protein or near the N or C terminus, as long as it's reasonably surface exposed for the following conversion. And then you can use formal glycine generating enzyme, which there are human versions as well as bacterial versions you can use to oxidize this cysteine into a formal glycine residue. And having this aldehyde there makes this amino acid unique from any other on the protein, and you can then react your molecules there. So, yeah, as I said earlier, I wanted to design it somewhere, my linkage somewhere where it wouldn't interfere with any of the properties of the antibody that I wanted to use in my in vivo model. So I engineered it to express the aldehyde consensus sequence of the C terminus of the antibody. And I have used in the lab several methods for converting the cysteine to an aldehyde. So one of my favorites to do nowadays is I have some stable cell lines overexpressing human formal glycine generating enzyme. And all then I have to do is transfect my plasmid into those cell lines, and the antibody it produces will be a majority aldehyde converted. And you can increase your yield by supplementing the media with some copper, since formal glycine generating enzyme requires copper for activity. Another method, which is a little easier and quicker if you're changing up your cell types, is just to co-transfect a human FGE plasmid and an antibody plasmid. And the yields will be very dependent on your transfection efficiency there for the yield for the conversion to the formal glycine residue. And finally, if for some reason, let's say you're working with a nuclear protein or something where it might be difficult to in vivo convert to the aldehyde tag just based on the localization of formal glycine generating enzyme, you can also just express your protein with the consensus sequence, still a cysteine, and use purified bacterial FGE enzyme to get the conversion. You will have to add a little bit of reducing agent and copper to get that reaction to go. But using those techniques, it was pretty straightforward to get an aldehyde bioorthogonal handle on my antibody. And then I used the hydroxynyl isopictet Spangler warhead. This was developed by David Rabuka at Catalent. And the advantage there is you can perform this reaction in mild acidic conditions, so like pH 5.5, and generate a stable carbon-carbon bond, which is really nice. But a lot of these reagents are not commercially available, which is the downside. So following that, I wanted to be able to get another bioorthogonal handle on the enzyme. And my original plan here was to actually use the formal glycine generating enzyme to get an aldehyde on this, the bacterial enzyme as well. But I actually found that when I expressed this, the consensus sequence, before I even bothered to convert it to an aldehyde, I tested it out in a few reactions, and I had actually made kind of a hot cysteine, or just a more reactive cysteine here. So I found that I was able to just alkylate that cysteine site specifically, not hitting any of the other cysteines on the protein, which saved me a couple of steps in converting to the aldehyde. So that was beneficial to me, that this aldehyde tag, I could I could site specifically label it without even converting to the aldehyde. But I conjugated on a strained cyclooctane so that I could then perform a quick reaction like we often do in the Bertozzi lab. And I will say this was very challenging with these two very large biomolecules. It took a really long time for this reaction to occur, and it was really difficult, if not impossible, to like drive to completion. Fortunately, I tested antibody-sialidase conjugates where there was 2 sialidases per antibody and 1 sialidase per antibody. And I found that having 1 sialidase, because it was catalytically active, was as good, if not a little better, at potentiating NK cell killing of cancer cells. So I didn't actually have to drive this reaction completely to conclusion, and I was able to use a mixture of diamodified and monomodified trastuzumab sialidase conjugate. And what these bioorthogonal tools allowed me to do, so this is an assay that I do in vitro, where we can mix antigen positive cell line and an antigen negative cell line. And when we treat with an antibody-sialidase conjugate, we hope to see selective desylation of the HER2 antigen positive cell line. So here, for example, is just using free unconjugated sialidase and using a lectin SNA to approximate sialic acid content on the cell. So with free sialidase, obviously there's no antigen specificity and both get desialylated at micromolar concentrations. But if we use the conjugate, T-Sia 2, we can get selective desylation at a little bit below the kD of the antibody. So only very little antibody really needs to bind to fully desylate these cells. And another advantage of having such a modular building block approach to synthesizing these antibody-sialidase conjugates is that I was able to assemble a bunch of control molecules really easily. So I was able to make an isotype antibody control that wouldn't target the cells, but would still give a huge half-life benefit to the sialidase when you inject it in vivo. I made an enzyme loss of function in the active site as a control just for like the biomolecules being there. And I also made some mutations to the antibody so it can't interact with Fc receptors on NK cells, macrophages, basically the innate immune system, and directly potentiate ADCC. So I had wonderful collaborators at University of Basel, and Michael Stanzak performed this mouse assay for me, where we used a HER2 positive syngeneic breast cancer model in mice, injected 4 times with the conjugate. We didn't really optimize that too much. It's just a procedure they typically do. And then looking at a tumor volume endpoint as a proxy for mouse survival, you can see that the T-Sia 2 conjugate really delays tumor growth and prolongs the mouse survival in this assay. And we were able to see that trastuzumab and a conjugate with a non-functional enzyme performed about equally. And interestingly, this model seemed to be completely trastuzumab resistant, even though there was the HER2 antigen on the cell line. Interestingly, the isotype control, which shouldn't target to the breast cancer cells, but will still have a really stable half-life and might be taken up by the tumors non-specifically, the isotype control actually did really well, which is actually kind of cool because this type of drug, if it were to be used as a therapy, would not have to, you wouldn't have to change or swap out the antibody for a specific type of cancer. It's more general. So although targeting seems to improve the desylation of the tumor cells, isotype control and just a stable sialidase injected can also be beneficial, which I thought was really cool. And then we found that in this model, which makes sense because it's a trastuzumab resistant model, so the function of the antibody as a therapy was not important. So if we deleted the effector functions of the antibody, it still performed equally as well as the normal antibody, T-Sia 2. And what this says is that it's really the therapeutic effect is really being caused by desialylation, which is really interesting because that's not something we see in the in vitro models. In our in vitro model systems, the antibody was always really beneficial. And we still have a lot of work to do figuring out the exact mechanisms and how desialation can be beneficial in lots of different tumor models, but we were able to do an experiment in mice that had sialic receptor knockouts, and we found that between the loss of function and T-Sia 2, there was actually no benefit without sialic receptors. So this more research needs to be done to confirm this, but it does support a sialic sialoglycan dependent interaction checkpoint mechanism for this model. And I really look forward to seeing where these molecules go in the future. And I would also like to thank the Holder Tosi Lab. Here we are pre-pandemic and post-pandemic. And in particular, Carolyn for her excellent mentorship and guidance on this project. Stephen for being here today and introducing me. And also my fantastic collaborators from the Loudly Lab. I showed mouse data from Michael and Natalia today, as well as some brilliant masters and undergraduate students who've worked with me, and David Rabuka from Catalent for some help with the aldehyde conjugation reaction. And thanks to you all for your attention.
Thanks, Melissa. It's really nice to see a talk with not one, but two different enzymes. We get both FGE as well as sialidase, so we get a double whammy here. Anya, do we have questions from the audience?
Yeah, so we have a question from Radka Snajdrova from Novartis. So Radka, would you like to unmute yourself and ask a question to Melissa?
Sure, thanks, Anya. And thanks, Melissa, for a great talk. I would perhaps have two questions. One would be, do you think there is any opportunity to use different targeted delivery than antibodies? And second, I don't know if I miss it, did you need to engineer the sialidase and maybe think of some extra stability for this therapeutic application?
Yeah, those are really great questions. I think, especially that first one, using different targeting moieties other than an antibody is a great idea, because when I designed T-Sia 2, I was kind of using ADCs and antibody therapies as my model. But it turns out that while those are very specific for drug delivery, if you're doing cell surface targeting, antibodies actually bind to a lot of cells, like epithelial cells and immune cells, through the Fc receptors. And so what I found, I mean, I have some tissue staining, is there was a lot of off-target effects throughout the mouse. So you can see, for example, a measure of sialic acid is in white here. And when I treat with T-Sia 2, the tumor is desialylated, but also there is some desialylation of other organs. So the mice were seemed very healthy, but I think more specific targeting could be done with different targeting proteins, like even an antibody or gnotins, like were mentioned earlier. And the second part of your question was engineering the sialidase, which I think is definitely the next step to take if this were to become a therapy, because I'm using the bacterial Salmonella sialidase, which I picked because it's very, very stable and it was really efficient at cleaving. I just wanted to see if this model would work in mice, but it would be much better to use maybe a human sialidase for immunogenicity reasons. And it would also be cool to engineer the sialidase so it's not very active, like really reduce the enzymatic activity of the sialidase so that you really need the antibody to localize it to a cell surface to see much of an effect. And that could also reduce these off-target effects I'm seeing. Thanks.
So Melissa, if I could follow up with another question regarding sialidase. So can you comment a little bit about the initial design that contained the maltose, the sugar binding domain on the sialidase, and then you chose the one without that sugar binding domain. You wanted it to be less specific, or what was the reason for those different designs?
Yeah. So yeah, the original design, the way they picked the sialidases, they before conjugating them all to antibodies, Hahn and Elliott did a screen where they treated cancer cells with antibody and free sialidases. And so Vibrio cholera sialidase is very efficient, and so they wanted something that would definitely work for their model. But once it was conjugated on, we realized the off-target specificity was really high, because not only was it binding to HER2 positive cells, it was also binding to any cells containing certain sialic acids. Just because Vibrio cholera sialidase itself has a pretty good KD, like it can work at 30 nanomolar on cells. So because I really wanted to get a little bit more, well, a lot more specificity in the mouse, I did a screen for one that was just a catalytic domain. And I think, which is relevant to the previous question, I think it could be better. I think it could be like basically a worse enzyme in this case would be better for my purposes.
Yeah, thank you for that. There is another question from Bernhard Geistanger from Merck. Do you see anti-drug antibodies against sialidase in mouse? Is multi-dosing possible?
We didn't look, because this is kind of an aggressive cancer model in the mouse, kind of a quick one. And it might usually take a couple weeks to see those antibodies, but I would guess almost certainly you would get antibodies against it, since it's a bacterial enzyme. So I think to prevent that in the future, we'd probably have to do some protein engineering using a human sialidase and trying to get that to be stable and maybe expressed as a fusion protein or something like that, just to reduce those possibilities, because I didn't test for it, but I'm almost positive it would be there.
Mm-hmm.